Again, today's lesson is on protein structure. There are 4 levels of protein structure that we want to. But before we examine those 4 levels of protein structure, I'd like to go ahead and revisit how we create proteins and a little bit about the bonding. In order for us to create proteins, if you remember from the last video, a covalent bond has to be formed between the 2 amino acids. And every time a new amino acid is introduced, then we're forming another covalent bond. Remember that covalent bond is called, in protein structure, a peptide bond. Try to grab a pen here. And that peptide bond is formed, if you remember from the first unit, dehydration synthesis reactions. In dehydration synthesis reactions, we remove a group and a hydrogen from another amino acid and form water. Okay, so we dehydrate the molecule. And you can see when we dehydrate the molecule, we're left with the remaining carboxyl group, and we're left with a remaining amino group. And so we're going to form a covalent bond between the carbon of one and the nitrogen of the other. So if you look down, it's showing you where that covalent bond is formed between the carbon and the nitrogen. Okay? Alright, so let's just go back and review a little bit as far as that was concerned. Let's move on. Okay? As we're linking those amino acids together from the first all the way to the last one, we have created a linear sequence of amino acids. And that linear sequence of amino acids is what's going to be responsible for determining our future protein structure. Because if we go back to the previous slide, one in the opposite direction, but if we go back to the previous slide, what we're going to see are interactions occurring between the side chain. Grab a pen. Remember these side chains, these are the R groups. Let me try it one more time. Okay, because remember, you have a central carbon, and off of that central carbon, you have a hydrogen, you have an amino group, you have a carboxyl group, and then you have what's called an R group. In the R group, these are the side chains. And if we move over to the next central carbon, or alpha carbon, you'll see another R group. And it's in these R groups is where many of these interactions are going to be taking place, which is going to determine our secondary structure, and which is going to help determine our tertiary structure. So let's go back. Alright, so this is showing you again the primary level. Primary level, again, that's just the linear sequence of those amino acids. Remember, we had 20 amino acids, and those amino acids arranged in different lengths and arranged in different ways is what's responsible for creating all the different proteins. So if we move to the secondary level of protein structure, there are 2 general forms. The 2 general forms are alpha helix and a beta pleated sheet. The alpha helix is a spiral structure, okay, and this one is common in globular proteins. The beta pleated sheet, as you can see, the sheet basically folds back on each other. Okay, I lost my pen, but anyway, but it's a folding process where the molecules fold back on each other. Now, the other thing is, both of these structures are held together by bonding. Okay, and Hydrogen bonding is a weak bond. If you remember, it's represented by a dotted line. And as you can tell, this hydrogen bond is occurring between what we call polar parts of the structure. So it's occurring between the hydrogen, which is generally carrying a positive charge from that nitrogen, and from the carbon you have this oxygen group, the remainder of a carboxyl group, and that's carrying a more of a negative charge. So those two charges are being drawn towards each other in order to create that hydrogen bond. Okay, let's keep moving. Our structure is tertiary level of protein structure. Now this structure is due to, again, remember how we talked about those side chains. So it's due to hydrogen bonding that's occurring between the side chains. Let me back up. Grab a pen. Okay, so this is showing you that hydrogen bonding occurring between the side chains. Some of the other interactions that are taking place is the hydrophobic interactions. Okay, hydrophobic interactions. Remember, hydrophobic is water fearing. So these methyl groups fear water, and they dimethyl, I guess, groups fear water, and they want to tuck themselves towards the inside of the molecule away from the water. So those are hydrophobic interactions that are taking place. Some of the other interactions that are taking place are ionic bonds. Okay, so we can have an ionic bond occurring between some of the side chains of groups. We also could have disulfide bridges where we have sulfur groups interacting and forming bonds between the sulfur molecule. If you look at the top, one of the other things that you want to remember about tertiary structure is the final folding and twisting that results in a globular molecule. So it's the final folding and twisting. So it can include alpha helices and beta sheets, or all alpha helices, or all beta sheets, or whatever, but it is the final folding and twisting. So, and all these different interactions that are taking place, and many of them are far away in the chain, so they're not just side by side, but these interactions occurring from one end of the molecule to the other that results in its final folding and twisting. So that's our tertiary structure. Usually our tertiary level of structure is where we are active. Say for instance, in tertiary structure, that's where you're going to have your active binding sites, for example, where a molecule is going to bind to its substrate in order to cause a change in whatever the substrate in order to create a product. The final level is your quaternary level of structure, and quaternary level of structure is due to interactions between tertiary molecules where you have either two or more molecules that are bonded together because of their interactions. And this one molecule that I'm showing you right now is hemoglobin, and hemoglobin is a quaternary protein, and it has 4 units, 2 alpha and 2 beta. One of the things, if you remember, and that's how important primary level of structure is, because for example, patients that have sickle cell anemia, there's 2 amino acids that have been changed in the primary level of structure in that linear sequence. And because those 2 amino acids have been changed, that causes the protein to fold differently once the oxygen has been released. And those people that have sickle cell anemia, have some serious issues. You know, once the cell sickles, those cells may be caught in the teeny tiny blood vessels and capillaries. But anyway, let's end up in this video just going back and talking about real briefly all those levels of structure. And we'll start with primary structure. Remember, that's just simply the linear sequence of amino acids, one amino acid following the next amino acid, then following the next amino acid, then following the next amino acid. You could look at them like beads on a chain, and then the string in between the beads representing the peptide bonds. Then we had two different types of secondary structure where the linear sequence may fold back upon itself and form a beta sheet, and in that you're going to find hydrogen bonding occurring between the oxygens of the carboxyl group of one and the hydrogens of the amino group of another. But this is again representing those hydrogen bonds occurring. We could also have an alpha helix. Again, the same interaction, hydrogen bonding occurring between hydrogens and oxygens within that molecule. Then when we have the final folding and twisting that results in a globular molecule, for example, this is our tertiary structure. So this is the third type of structure. And in this structure, you can see that we actually have some beta sheets as well as an alpha helix. And then finally, the quaternary structure, where we have more than one of these tertiary molecules. So here's two, and then they're showing you two more on the back side. But anyway, those are our levels of protein structure, and we will talk to you again later in the semester. Have a good class.